to talk about the cell structure. So let's jump right into it. We're going to start with eukaryotic cells and organelles. So, cells, I'm going to read from the book. Cells, what an original way to start a biology book. Oh well, there are two types of cell, prokaryotic cells and eukaryotic cells. First piece of information, we have two types of cells, prokaryotic cells and eukaryotic cells. Eukaryotic cells are stuffed full of organelles, all the tiny bits of bulbs that you can only see in detail with fancy microscopes, and you need to know about all of them. So basically, the saying like the eukaryotic cell is made up of many different organelles which can only seen by very sophisticated microscopes. Prokaryotic organisms are prokaryotic cells, i.e., they have single cell organisms. They're basically saying like prokaryotic cells are single cells, so they only exist as one cell. So a prokaryotic cell can only be one cell, one single cell. Eukaryotic organisms are made up of eukaryotic cells. Eukaryotic means that eukaryotic organism can also be multicellular, like made up of many different cells, like your body is made up of millions of cells. Eukaryotic cells are complex and include all animal and plant cells. So examples of eukaryotic cells, we can say animal cells and plant cells. Prokaryotic cells are small and simpler, e.g. bacterials. So from this we can conclude that prokaryotic cells are smaller than eukaryotic cells. Both types of cell contain organelles. Organelles are part of the cell. Each one has specific function. So both prokaryotic and eukaryotic cells have organelles, and those organelles have many different functions they carry out. Eukaryotic cells are generally a bit more complicated than prokaryotic cells. You've probably been looking at animal and plant cells diagram for years, so hopefully you'll be familiar with some bits and pieces. So they're saying eukaryotic cells are a bit more complex than prokaryotic cells because they have like more organelles, they carry out more functions, whereas the prokaryotic cells are simpler with less organelles that carry out less functions. So we have the diagram of an animal cells. Unfortunately, I can't show you the diagram on the stream right now. So animal cell, we have the plasma membrane, we have the rough endoplasmic reticulum, we have the nucleus, the nucleolus, the smooth endoplasmic reticulum, the lysosome, the ribosome, the nuclear envelope, the Golgi apparatus, the cytoplasm, and the mitochondria on the cell membrane. If you can find an image of a cell, of an animal cell, you can find it online and like follow up with the different parts of the cell. Plant cell. Plant cell have all the same organelle as the animal cell. So they're saying the plant cell has all the same organelles as the animal cell, but with a few added extra. So they have the same with some few more organelles which the animal cell doesn't have. A cell wall with plasmodesmata channel from exchanging substance with adjacent cells. So they have a cell wall, and the cell wall has plasmodesmata, and the cell wall basically channels the exchange of substances from one plant cell to another, like it controls what enters into the cell and what comes out of the cell. A vacuole compartment that contains cell saps, and of course good old chloroplast. Basically the chloroplast is what carry out the photosynthesis, like is the site for photosynthesis, and it has like chlorophyll, like it gives the greenish aspect of the plant's leaf. eukaryotic cells and organelles. Okay, so we have a table. The giant table contains a big list of organelles. You need to know the structure and functions of them all. Sorry, most organelles are surrounded with membrane, which sometimes causes confusion. Don't make the mistake of taking that diagram of an organelle as the diagram of a whole cell. They are not cells, they are part of the cells. So we have a table. Also can show you the table, but basically the table have like the diagram of each organelles with their functions and structures. So they're saying like most organelles are surrounded by membranes. Don't confuse them for cells. They are still organelles. So the first one we have plasma membrane. Under the plasma membrane, it shows we directly have the cytoplasm. Description: the membrane found on the surface of animal cell and just inside the cell wall of plant cell. So the plasma membrane is the surface of animal cells, and in plant cells it is found directly under the cell wall of plant cells and prokaryotic cells. It's made mainly of lipids and proteins. So we can find plasma membrane on animal cells, plant cells, prokaryotic cells. It is made up mainly of liquids and proteins. Functions: the function of the plasma membrane, it regulates the movement of substances into and out of the cell. It also has receptors molecules on it, which allows it to respond to chemical like hormones. So they say it regulates the movement of substances in and out of the cells, and it also has receptors on it. Next, we have the cell wall. Description: a rigid structure that surrounds plant cells. It's made mainly of the carbohydrate cellulose, and the function is to support the plant cell because the cell wall is only found in the plant cell. Next, we have the nucleus. Description: a large organelle surrounded by a nuclear envelope double membrane which contain many pores. The nucleus contains chromatin and often a structure called nucleolus. Chromatin is made from proteins and DNA. Controls the cell activities. The pore allows substance, e.g., RNA, to move between the nucleus and the cytoplasm. The nucleus makes ribosomes. So basically, the nucleus has pores that allow substances to move from the nucleus out to the cytoplasm. Usually the RNA, because like the nucleus is the site for like RNA production. No, pardon. The nucleus makes ribosomes, so the nucleus is the site for ribosome production. Next, we have the lysosome, a round organelle surrounded by a membrane with no clear internal structure. Contains digestive enzymes. These are kept separate from cytoplasm by the surrounding membrane and can be used to digest invading cells or to break down worn out components of the cell. So, the lysosome are round and surrounded by a membrane to separate them from the rest of the cell. They contain digestive enzymes which helps the cell to break down foreign cells that get into it or to break down worn out components of its own, like worn out components found in the cell. So next we have ribosome. We have the small subunit and the large subunit. Description: a very small organelle that either floats free in the cytoplasm or is attached to the rough endoplasmic reticulum. The site where proteins are made. Ribosomes is the site where proteins are made. Next, we have rough endoplasmic reticulum. Description: a system of membrane enclosing fluid filled space. The surface is converted with ribosomes, flood and process function, flood and processes proteins that have been made at the ribosome. Smooth endoplasmic reticulum. It's similar to the rough endoplasmic reticulum but with no ribosomes. Function: it synthesizes and processes lipids. Next, we have the vesicles, a small fluid filled sac in the cytoplasm surrounded by a membrane. The vesicle, its function is to transport substances in and out of the cell via the plasma membrane and between organelles. Some are formed by the Golgi apparatus or the endoplasmic reticulum, while others are formed at the cell surface. Next, we have the Golgi apparatus. Description: a group of filled sac, flattened sac. Vesicles are often seen at the edges of the sac. Okay. Function: it processes and packages new lipids and protein. It also makes lysosomes. Next, we have the mitochondria. They usually description they are usually oval. They have a double membrane inner one folded form structure called crystal inside the matrix, which contain enzyme involved in respiration, but sadly no new ravis. I don't know about that. Alright. The function, the site for aerobic respiration where ATP is produced. They are found in large numbers in cells that are very active and require a lot of energy, generally muscle cells. Next, we have the chloroplast. Description: a small flattened structure found in plant cells. It's surrounded by a double membrane and also have membrane inside called thylakoid membrane. These membranes are stacked up in some parts of the chloroplast to form grana. Grana are linked together by lamellae, thin flat piece of thylakoid membrane. Function: the site where photosynthesis takes place. Some part of photosynthesis happen in the grana and other parts happen in the stroma, a thick fluid found in the chloroplast. Next, we have centriole. Description: small hollow cylinders containing a ring of microtubules, in bracket tiny protein cylinders. Functions: involved in the separation of chromosomes during cell division. Next, we have the cilia. Description: small hair-like structure found on the surface membrane of some animal cells in cross sections. They have an outer membrane and a ring of 9 pairs of protein microtubule inside, with a single pair of microtubule in the middle. The microtubule allow the cilia to move. This movement is used by the cell to move substances along the cell surface. Next, we have the flagellum. Flagella on eukaryotic cell are like cilia but longer. They stick out from the cell surface and surround the plasma membrane inside like cilia too. Two microtubules in the center and 9 pairs around the edges. Function: the microtubule contract to make the flagellum move flagella and use like outboard motors to propel cell forward, e.g. when a sperm cell swing. Okay, now we have some questions. Question 1, describe the function of a vesicle. They said the vesicle has a membrane and it basically like transports substances in and out of the cell. Mhm. Question: so, how does the structure of the rough endoplasmic reticulum differ from the smooth endoplasmic reticulum? Question two. Basically, the rough endoplasmic reticulum has ribosomes attached to it, while the smooth endoplasmic reticulum does not have ribosomes attached to it. And also in the function of the smooth endoplasmic reticulum is like it deals with like lipids and carbohydrates, whereas the rough endoplasmic reticulum mostly deals with proteins. So question three, what is the function of the chloroplast? So we know the chloroplasts are found in plant cell, and like that is the main place where photosynthesis is being carried out because it contains the chlorophyll that makes the leaf green. So question four, what is the function of the centriole? You know the centriole is in plant cell and is mostly used during cellular division to like split the cell into two, each of them going at different ends of the cell. So we have question five. Name one organelle found in an animal cell. This one should be pretty basic. We have a lot of organelles in the animal cell. So one of them we can say the nucleus, it's an organelle, the mitochondria, the Golgi apparatus, like basically everything we just saw above are organelles. So now we have exam questions. Question 1. Give 4 things commonly found in plant cells but not in animal cell. 4 marks. Commonly found in plant cell but not in animal cell. So firstly we know the plant cell has all organelles that the animal cell have. But now the plant cell has more organelles that the animal cell doesn't have. So they ask for 4. So firstly, we can talk about the cell wall. We know the plant cell has a cell wall, but the animal cell doesn't have the cell wall. Secondly, we can talk about the chloroplast. We know the plant cell has a chloroplast to carry out its photosynthesis, whereas the animal cell doesn't have a chloroplast since it doesn't need to carry out photosynthesis to get its food. Fourthly, what organelle can we talk about? Fourthly, we can talk about like structural differences. We can talk about a larger vacuole. Plants have a larger vacuole to store more carbohydrates that they produce in their vacuoles, whereas the animal cell have smaller vacuoles. So for food difference, what can we say for the food difference? Mm, for difference, let me think for a minute. All right, for now we're going to leave it at three differences. Let's go to the next question. Question 2, identify these two organelles from the description as seen in the electron microscope. So they're going to describe two organelles and we're going to tell which organelle it is from the description. Okay, number 1, an oval shaped organelle surrounded by a double membrane. The inner membrane is folded and projects into the inner space, which filled with grainy material. This is surely the mitochondria. We saw above that the mitochondria has a double membrane. The inner membrane is folded, projecting inward. So this is the mitochondria for sure. The second one to say a collection of flattened membrane sac arranged roughly parallel to one another. Small circular structure are seen at the edge of this sac. Small circular structure are seen at the edge of this sac. Structure called flattened membrane sac. Flattened membrane sac arranged roughly parallel to one another. This should be the chloroplast because they say a collection. We know in the chloroplast we have a collection of thylakoids, which are flattened membranes sac, arranged roughly, that are roughly arranged parallel to other because they stack up on one another. To one another, small circular structure are seen at the edge of the sac, and they are usually found at the sage, at the edges of the sac, and they are linked by filaments. So that should be the chloroplast. B. Okay. B. State the function of the two organelles that you have identified. The first one we identified was the mitochondria. We know it's the powerhouse of the cell, the site for aerobic respiration, ATP production. And the second one was the chloroplast. We know it's the main site for photosynthesis. So we've answered the questions. What do they say next? That's enough talk of fluid filled sac for my liking. Scientists these days, organelles is very pretty sounding name for all those blobs. Actually, under the microscope, some of them are really quite fetching. Well, I think so. Anyway, but then my mate find sheep fetching, so there are no accounting for taste. Anyways, you need to know the names and function of all organelles and also what they look like. So we need to know the shape and function of all organelles and what they look like. Organelles working together. After that, endless list of organelles. After that endless list of organelles, you might need a few minutes to regain consciousness. Then you can read this lovely page about how they work together to produce proteins. And there is some stuff on cytoskeleton too. Whoop. Okay, so we have a graph for like protein production in cells. Okay, one, proteins are made at the ribosome. Proteins are made at the ribosomes. That's the first point. Proteins are made at the ribosomes. Two, the ribosomes of rough endoplasmic reticulum make proteins that are excreted or attached to, to the cell membrane. The free ribosome in the cytoplasm make protein that stays in the cytoplasm. So, the saying that the rough endoplasmic reticulum makes protein that are excreted out of the cells or attached to the cell membrane, whereas the free ribosomes moving in the cytoplasm produce proteins that stay in the cell, in the cytoplasm. So that was the second point. Now, the third point, new proteins produced at the rough endoplasmic reticulum are folded and processed, e.g., sugar chains are added in the rough endoplasmic reticulum. So, the proteins produced at the rough endoplasmic reticulum are folded and processed, like some sugar chains have been added to them. Alright, that was the third point. The fourth point, then they are transported from the ER to the Golgi apparatus in the vesicle. Then they are transported from the endoplasmic reticulum to the Golgi apparatus in vesicles. So basically, the vesicles as a means of transport, like to transport stuff in the cells. So the vesicle transports the proteins that have been produced and processed from the endoplasmic reticulum to to the Golgi apparatus. That was their fourth point. Their fifth point. At the Golgi apparatus, the protein may undergo further processing, e.g., sugar chains are trained or more are added. So after being transported by the vesicles to the Golgi apparatus, more processing is undergone by adding or removing some sugar chains. That was the fifth point. The sixth point. The proteins enter more vesicles to be transported around the cell, e.g., glycoproteins found in mucus move to the cell surface and are secreted. The protein enters more vesicle to be transported around the cell. So basically, after the processing in the Golgi apparatus, the proteins are sent into other vesicles to be transported around the cell, out of the cell, on the cell. That was your 6th point. So here we have a diagram of protein production in cells, but unfortunately I can't show you the diagram right now. So but the diagram basically explained what we just saw, but on the diagram, from the ribosome to vesicles to Golgi apparatus to other vesicles, like it's the same thing I just said. The organelle in cells are surrounded by the cytoplasm. Like all organelles in the cell are being surrounded by the cytoplasm. The cytoplasm is more than just a solution of chemicals, though it's a good network of protein thread running in it. These protein threads are called cytoskeleton. In eukaryotic cell, the protein threads are arranged as microfilament, small solid strand, and microtubules, tiny protein cylinders. So basically now we have a diagram of the Golgi apparatus, the microfilament, the microtubules. Okay. The cytoskeleton has four main functions. Functions of the cytoskeleton. One, the microtubule and microfilament support the movement of cell organelles, keeping them in position. Microtubules and microfilament support the movement of organelles in the cell, keeping them in position. That's the first point. Two, they also help strengthen the cell and maintain its shape. The cytoskeleton helps to strengthen the cell and maintain its shape. That was the second point. Three. As well as this, they, they are responsible for the transport of material within the cell. For example, the movement of chromosome when they separate during cell division depends on contraction of microtubules in the spindle. Okay, that's cell division. We're going to do cell division for another day. So, number four, the proteins of the cytoskeleton can also cause the cell to move. For example, the movement of cilia and flagella is caused by cytoskeleton protein filament that run through them. So, in case of single cell that have flagellum, e.g. sperm cell, the cytoskeleton propels the whole cell. Alright, now we have prokaryotic cell. You need to be able to compare and contrast prokaryotic cell and eukaryotic cell. This being orange table should help. So we have a table. On the left side, we have prokaryotic cells and we have eukaryotic cells. So I'm gonna give you the difference between prokaryotic cell and eukaryotic cell. Prokaryotic cell, extremely small cell, i.e. less than 2 micrometers of diameter. Eukaryotic cell, larger cell, usually 2 to 200 micrometer in diameter. Prokaryotic cell, the DNA is circular. Eukaryotic cell, the DNA is linear. Prokaryotic cell have no nucleus. DNA is free in the cytoplasm. Eukaryotic cell, nucleus present, DNA is inside the nucleus. Prokaryotic cell, cell wall made up of polysaccharides but not cellulose or chitin. Eukaryotic cell, no cell wall in animal cells, cellulose cell wall in plant cell, or chitin cell wall in fungi. Prokaryotic cell, few organelles, no mitochondria. Eukaryotic cell, many organelles, mitochondria present. Prokaryotic cell, small ribosomes. Eukaryotic cell, larger ribosomes. Example of prokaryotic cell, we have E. coli bacterium. Example of eukaryotic cell, we have human liver cell. All right. So one, prokaryotic cell like bacteria are roughly and a tenth size of eukaryotic cell. So that talking about the size is like prokaryotic cell like bacteria are a tenth the size of a eukaryotic cell. So they're way smaller than the eukaryotic cell. So this means that normally microscopes aren't really powerful enough to look at their internal structure. Oh. The diagram shows a bacterial cell as seen under an electron microscope. So we have the diagram of a bacterial cell. We have DNA in bracket bacterial chromosome. We have flagellum, tail used to propel the cell. We have ribosomes. We have plasmid, ring in bracket ring of DNA. We have the cell wall. We have the plasma membrane, cell surface. Okay, so now we have more questions. Yeah, we have more questions. Yeah, we have more questions. Okay, let's try doing this question from what we just read. Question one. Give two structures that you might find in prokaryotic cell, but not in an animal cell. But not in an animal cell. Two structures you might find in prokaryotic cell, but in an animal cell. So for this question, I'll say the flagella, the tails it uses to swim, because the animal cell doesn't have flagella to swim. It has the cytoskeleton, yes, which has filaments and microtubules, but not the flagella. So that would be my first different structure I can find in prokaryotic cell, but not in animal cell that I'm gonna use. Secondly, Secondly, circular DNA. Prokaryotic cell have circular DNA, but animal cell we can't find circular DNA in animal cell. We have linear DNA. So that would be the answer for question one for me. Question two. An undefined cell is found to contain mitochondria. Is it prokaryotic or eukaryotic? This one is pretty easy. An undefined cell is found to contain mitochondria. Is it prokaryotic or eukaryotic? For this one, I'm gonna 100% go for eukaryotic. It is eukaryotic because eukaryotic cells, like animal cell and plant cell, both have mitochondria. Okay, now we have exam questions. Question one. Some mucus secreting cells were immersed in a solution of radioactive amino acid. Some mucus secreting cells were immersed in radioactive amino acid. Every 5 seconds, some of the cells were removed and their organelles were separated and analyzed. The radioactivity in the different organelle was measured for each 5-second interval. When answering the first two questions below, use organelle from this list: Golgi apparatus, ribosome, rough endoplasmic reticulum, vesicle. In which of these organelles will you expect radioactivity to appear first? Explain your answer. So, we have the Golgi apparatus, ribosome, rough endoplasmic reticulum, and vesicle. Hmm. Rough endoplasmic reticulum. Mhm. Radioactive substances. So, for this question, I'm gonna go with the rough endoplasmic reticulum. Let me explain why. Because during protein production, we start at the rough endoplasmic reticulum where the ribosomes are found, and radioactivity usually causes mutation, and mutation are usually seen through proteins. So from this, I will expect the first place that should be affected by the radioactivity to be the place where the protein have been produced at the rough endoplasmic reticulum. That would be my explanation. And B, after 5 minutes, the Golgi apparatus had become radioactive. Yeah, so that basically saying like from the previous question, the Golgi apparatus can't even be the answer. After 5 minutes, the Golgi apparatus had become radioactive. Which other organelle will be radioactive by this time? So basically, if we still stay on the concept of protein synthesis, everything before the stage of the Golgi apparatus in the process of protein synthesis should be affected. So after 5 minutes, the Golgi apparatus has become radioactive. Which other organelle will be radioactive by this time? The rough endoplasmic reticulum, the vesicles. It depends also on the vesicles because there are two stages where the vesicle transport the protein that have been produced. So I'm gonna keep that aside for now because we don't know which vesicle. So rough endoplasmic reticulum and ribosomes. Those would be my two answers. C. The researchers were particularly interested in animal cells vesicle. What is the function of the vesicle? 1 mark. Basically, the function of the vesicle is to transport substances within the cell. In the case of protein synthesis, it transports the proteins that are being produced and processed. Question 2. Give three functions of the cell cytoskeleton. Function of the cytoskeleton. It, it helps to maintain the cell's shape and structure. It helps in the movement of organelles between the cell and also help in the movement of the cell. Those will be the three answers for the three functions. So a cell without a nucleus, that can be right. It's like push without bags. Prokaryotic are way older than eukaryotic. In fact, most cellular biologists think that mitochondria and chloroplasts are remnants of ancient prokaryotic cells that live inside the first eukaryotic and eventually just became part of them. Mad. So that will be it for today. Thanks for watching this video. If you like what we did today, you can subscribe for more. I'll be doing this little biology studying answering question type of video every day. We're gonna go live every day doing this biology stuff. So if you like the video, drop a like. You can comment the issue you had with the video or what you didn't understand. Yeah, and subscribe if you want to follow for more. See you guys in the next one. Bye.